Okay. So now it's recording. So, okay, Katja. So everyone, thank you. So everyone should thank to Katja. So everyone buys coffee to Katja later on. If you can finish all those coffees, good. Anyway, so again, thanks to Katja for providing recording. Here is what so here is how the exam looked first. I will tell you briefly. So again, there are many bonus questions, so you should be fine, which you don't have to respond. It's 100 marks, and you have you have 3 sections. Section 1 or part 1 is 50 points total. That means that literally, and these are only multiple choices. So answering those easy questions, you can pretty much get half of them of the marks. And that question, so that section, 50 points, has 25 multiple choice questions, which means multiple choice question is worth 2 marks. If something has 2 correct answers, I will give you 1 mark. So, and I will come later to the second 2 sessions, like section 2 and 3, but let's go first to multiple choice section. So again, what you need to know, what will be on multiple choice are microbial eukaryotes, so those groups that we talked about. So you need to know their basic characteristics, their landmarks. That's basic. So which one has 2 nuclei, which one is parabasal body, which one has cytoplasmic sacs, etc., etc. So again, not in details, of course, but you have to know those characteristics because there will be matching. Then you need to know, we are again with microbial eukaryotes. So I do want you to know basic characteristics of amoebas, both types, and that nice little creature that has tons of cells, whose name I won't tell you because then I would tell you the name of the answer on the question. So that's another matching point. Then you have to understand significance of viral evolution, like what is, like when we talk about viruses, like there are a few slides at the end that talk about viral shunt in soil, and one specific enzyme that only viruses have is called reverse transcriptase. So you have to understand what is why are those 2 things are hint to understand significance in viral evolution. Then you have to know a few species. Again, there will be no specific, like when we talk about the biodiversity of microbes in various habitats, we mentioned a few species that do like plastic degradation, chlorine degradation. Then some of them are significant in corrosion. So you have to know those organisms, not to write, of course, just to recognize. The difference between genomics and genetics, it is on one of the slides when we talk about analysis of microbial biodiversity, like what is genomic interested in, what is genetics interested in. Then you have to understand all 3 theories about viral evolution: virus early, regression hypothesis, and escape genes, like what each of them says. Then again, we are at the evolution of viruses. Those 2, we talked about 2 hypotheses on viral divergent and convergent evolution. So you have to know what divergent evolution theory says, what convergent evolution says. You need to understand basics of biofilm formation. So when we talked about biofilm, there is one specific molecule that bacteria that form biofilm have. The molecule is called cyclic DGMP, and it's made by an enzyme called D-guanylyl cyclase. So read that mechanism.
Sorry, Maria, can you speak a little louder? I couldn't hear.
D-guanylyl cyclase makes cyclic DGMP that is involved in biofilm production. Read that mechanism. Okay. Then you need to know which methods are culture dependent, which one are culture independent for analysis of microbial biodiversity. Read about invasion of virus in the cell. There is specific order that viruses follow when they're invading host cell. Then in terms of type 6 secretion system, you have to, so read Nora's lecture. Again, she made questions. So you need to know some basic things about type 6 secretion system. So and you had that similar, you had the question, there was one bonus, actually 2 bonus questions on quiz 3. So now it's in main body. So just understand that strain A, who wins? Know how to recognize what is type 6 secretion system, like when you see it. So those are all bonus questions. And then in terms of, no, actually, no, this was not bonus. This was, sorry, this was main question, now bonus questions. So great oxidation event, when it happened, how it happened. Banded iron formations, very important.
Sorry, can you say that again?
Banded iron formations, like Dr. Kornhauser lecture. Hint drop in methane. Dr. Kornhauser was talking about that. And then another hint, a high chromium concentration. So some indicators that Kurt was talking about. And again, great oxidation event. That's what Kurt really likes. In terms of Brittany's lecture, there are 2 bonus questions. So you have to know, read briefly what are some genuses present in crabs, if you are interested, and know some specificity of one of these genes, like amino acid related. And that's all for part 1. Okay. So that would be, so you have 25 questions plus bonuses, multiple choice. Then part 2 is, yes.
Sorry, could you please just repeat what the second bonus questions?
Amino acid present in one crab gills, yes.
Were these lectures on YouTube?
Yes, yeah, I posted it like 2-3 days ago. Yeah. But again, this is for you, just, you know, if you want, which I would really like, but that won't give you, like, you get bonus, you cannot get 110, you can get only 100%. Part 2 is little hard. So part 2 is the hardest here. So part 2 has fill in the gap. And what I did this time is there is only one line, and then in bracket it says how many words you should put. So you don't have to, so don't pay attention on, I mean, there is only one line, but I don't know if printing is bad or something, like it should be only one line, is my point. So there are 4 questions that will be fill in the gap, and these questions are quite intensive. So my expectation is that you will finish multiple choice in 15 minutes, and then the rest will spend on next 2 sections. So fill in the gap, first question carries 10 points. So the second session is like 40 points total. So that's pretty intensive, but the first one is 50 points. So 50 points you can get for 15 minutes, and then we will distinguish between people who should get A plus, C minus. So again, there were some, I got some emails being like, there should be a word bank. No, this is third year. This is third year course. If you reach third year of your classes. So word bank might exist in 107, 108 when you are first year, when you don't really have knowledge, you are third year course. So you have to know how to think. If you don't, okay, that's fine, you will lose marks, but that I have to be able to distinguish somehow people who get A plus and people who get C minus. So in terms of first question, it is about viruses. So you need to know what is virus, like a key hint, it's not cell. That's a key point. Which nucleic acids are present in viruses? How it's called, that thingy that surrounds nucleic acid in viruses? What is the viral lifestyle? So, and there are 3 words there. So you will see. Then some viruses have some additions that are not encoded by their own genome, so know that. Then know decorations on viral cell, and know about bacteriophages and 2 types of bacteriophages that we have. Then second question also carries 10 points, and it is about primary and secondary endosymbiosis. So you have to know which two organelles are a result of primary endosymbiosis and how they contributed to secondary endosymbiosis. So you need to know what are which organisms we mentioned specifically that are a result of secondary endosymbiosis. And you need to know which organelles are doing what, like those two organelles that I won't tell you, but you should know. One is doing one thing, the other one is doing another thing, and that's very important to understand. And you need to know type of ribosomes that bacteria have and how are those ribosomes connected to primary endosymbiosis, how they contribute to the primary endosymbiosis. So a hint, we have ATS. That's a hint. 80. Bacteria have something different. You need to know what's the name of those specific organelles that are present in. So technically there are three types of organelles that we talk about when we talk about primary endosymbiosis. So one of them is very specific. And again, I can't tell you because I would tell you the answer to question, but you need to know. They're also related to the evidences that support primary endosymbiosis. Question 3, also 10 marks. It's about interactions between, microbes in different habitats. So we talk about those three types of interactions. Again, I won't tell you because then I will tell you answers. So when we talked about diversity of microbes in various habitats, we talked about three types of interactions. One of them involves fungi, and so you have to know that. That's what you will be asked. And what is specific about them, like what is each of them doing? So that's a hint. Fourth one, and the last in this section, is also 10 points, and it is about microbiome, ruminant microbiome, like what are dominant organisms, archaea, bacteria, those kind of things. I like that one specific organism that I mentioned in that lecture, so know that organism and know that very cute example of how we can help our ruminants just by feeding them with something from gut. So how we call that something from gut. Part 3 is concepts and definitions, and there are 5. And each of them, so it's total 10 points, but there are 5 definitions. Again, I can't tell you which definitions are, but each of them is 2 points, which means, so it's not a lot of points, but I can't tell you exactly what you should write, but just make sure you fill up the space that I gave you. But like, so I leave exactly enough space, and I will give you one extra piece of paper on the back so you can, write in case somebody really writes big letters like myself. But that's what is expected. So if you don't write one word, if you see like this large space. So you need that there are again 5 definitions. You probably understand I can't tell you what is definition, but I can tell you that I will ask something specific about methodology. Like there are 2 methodologies that I will ask specifically, non-culture culture independent, and I will ask something related to hydrocarbon degradation and something about recent findings in viruses.
Sorry, can you do that last one?
Recent findings about significance of viruses.
What's the second point of the
Hydrocarbon degradation. So again, you should definitely study. You have like 6-7 lectures. That's not a lot. So read questions carefully. You have 3 hours. So that's way, I did exam in 20 minutes. So we have a rule that says like you make exam, you wait 1 day and then you do exam, and then whatever you take to do exam, students need 3 times more. So if I took 20 minutes, you technically, if you study, you should be able to finish in 1 hour. So that's my, again, if you studied. If you did not study, then you can really sit here 5 hours, you won't do well. So some people sometimes need extra time to think. I understand that, but I am different. If I study, I can do it very easy. if I didn't study, I can sit here 10 hours. I'm not going to recall anything. So that is all for this section. I will now, because it's, as I said, it's quite fast lecture, so I will stop recording and return laptop to Katja. And I am going to go back to my office to get my to get your quizzes, and I will sit in Remedy Cafe if you want to come and see your quiz trees. So I might be there around 1:15 because I have to go to the office to try to find something. But if you guys see me there and I don't see you, please approach me because I don't really remember all of faces. I have some landmarks here, so that I will definitely memorize. So that's all from me. Good luck on Monday. I want all of you to study. So boost up because your GPA is my GPA now. So run. See you later. Thank you, guys. You don't like me, right? Oh God, that's kind of true. I have to tell this to my husband. He was saying that you're just pretending. I was like, no, they like me. Thank you so much.